Hello, my name is Sin Kim, and I'm here to talk about my search for associations between polygenic risk for major psychiatric disorders and early adolescent white matter developments. First, I'll briefly describe the big picture behind my study. Adolescence is a key developmental period during which many psychiatric disorders emerge. And many of those disorders are associated with defects in white matter structure. And that white matter undergoes significant development during adolescence. In general, white matter increases in this period due to increase in myelination and axon densities. And importantly, this increase is nonlinear, meaning there's a change in slope during adolescence. Thus, my goal is to find factors that predict this change in trajectories. For this study in particular, I examined baseline white matter and genetics. Why these factors? Well, it is well known that genetic factors play a large role in psychiatric disorders and white matter structure. But interestingly, gene expression can also change during adolescence, which means some genetic effects on white matter may be specific to adolescence, and those may trigger changes that eventually lead to psychiatric disorders. Thus, I hypothesized that genetic risk for psychiatric disorders may alter adolescent white matter development. To test this hypothesis, I used the ABCD study, a large longitudinal multimodal dataset, and based on GWAS results from even larger datasets, I estimated each subject's polygenic risks for various psychiatric disorders. Then I tested for associations across genetic risk, white matter structure, and symptom scales. This three-way analysis was repeated with longitudinal change scores for white matter and symptoms. As a disclaimer, although ABCD dataset includes over 10,000 subjects, including only unrelated subjects with quality-controlled longitudinal data, half this number. Furthermore, because polygenic risk score loses predictive power across different ancestries, only European ancestry subjects were included, which halved the sample size once again. Hopefully, there will be more GWAS in the future that examine diverse populations. As for my covariates, socioeconomic variables, which include combined household income and highest parent education, were not included because there may be colliders depending on how we assume they relate to polygenic risks. However, including them did not change results anyway. Also, DTI confounds were included because even if they were mediators, we're more interested in direct effect anyway. And now for the results of these analyses. For most psychiatric disorders, polygenic risks is associated with symptom scales, meaning GWAS from adult population do replicate in adolescents. But interestingly, only anxiety and depression polygenic risks were associated with changes in symptoms. And unfortunately, when including all the necessary covariates, I was not able to find strong associations between polygenic risks and white matter structures, or change in white matter structure. Thus, the results did not support my hypothesis, and I could not continue with my mediation analysis. As for other relations, while white matter was not associated with baseline symptoms, change in a couple of white matter tracts were associated with change in ADHD symptoms, which is kind of odd because ADHD polygenic risk was not associated with change in symptoms. In addition, in post-hoc moderation analysis, I found curious interactions between polygenic risks and white matter. Polygenic risk negatively moderated baseline to change association for bipolar and schizophrenia, and positively moderated baseline white matter to symptom associations, while negatively moderating white matter change to symptom change for ADHD. I am not sure how to interpret these results, but they may be worth future investigation. In summary, my first clear result is that polygenic risk and early adolescent symptoms are associated for ADHD, anxiety, and depression. Thus, the GWAS from adult populations do replicate in adolescents for these disorders. A very recent paper from another group also found similar results, which further supports this finding. Another clear result is that polygenic risks for anxiety and depression are associated with emergence of the symptoms. However, to determine neurobiological mechanism behind this, we will need to investigate a wider variety of potential factors than just white matter structure. An important limitation of this study is that the most significant growth of white matter between age 14 and 16 has not yet been captured in the ABCD data set. In addition, with only 2 longitudinal time points, we cannot use more powerful longitudinal statistical methods. It will be worth revisiting this hypothesis, my hypothesis, with a later ABCD data release. Thank you for listening. You can find more results and my full reproducible code at this GIN repository.